Well, we've got a lot we can talk about. How many minutes do we have for discussion? Just till quarter after, so 25 minutes. Okay, got it. That's great. And we have Kyle Myers joining us from the FDA for the discussion, who's a key person in CDRH's Office of Science and Engineering. So I just a quick comment. Before I ask the first question, and then I hope, seeing the audience, I'm sure you guys will have a lot of interesting comments and questions. But I sort of grew up trying to combine data from a medical student on, and listening to you guys, you would think you can just sort of apply some magic and poof, you've got stuff that you can analyze and make sense of. But somebody who Chuck Jaffe knows very well, when I was a third-year medical student, told me when I was looking at some disparate data, you can't make chicken salad out of chicken blank. And are we now in an era where you can take data that has different meanings for the same terms and collect it in different ways under different circumstances and put it together and make sense of it? And if not, what is the role of data curation, and how much of your effort needs to go into this? I have frequently used a term I love that came the New York Times, called the data janitor. I've since learned that a lot of informaticians don't like that I use that term. But I actually worked as a janitor. I think it's a noble profession. So I think cleaning things up is a really good thing to do. But am I mistaken? Can you actually work magic now, or do you need to have curation? And if so, can you describe a little bit about what's involved?
I guess the magic depends on the quality of the ingredients to a certain extent, and I think that there is an issue about understanding what is the quality of your data and in what ways is it of poor quality. And that's why I was trying to get across the difference between systematic issues, particularly systematic missingness. I think things like migration, if people are using between one healthcare setting and another, for example, or if people are doing mobile health trials, app fatigue. I've downloaded this app, I've used it for 3 days, and now I'm not going to bother anymore. It's not a conscious decision, but it might be a form of informative censoring. So there's a whole load of that stuff that you've got to get to grips with. I think the second is that there is a lot of noise and variation in the coding, but actually some of the concepts persist over time. Perhaps the best example, I think, it comes from myocardial infarction. We're on our third universal definition of myocardial infarction. Now, for those who are not from cardiovascular disease, you're probably surprised that there is a third universe, which presumably meant there was a second and a first, and almost certainly means there'll be a fourth. Now, actually, the trials that you ran on thrombolysis from the 1980s didn't use any universal definition. They used a pragmatic definition, and that information about thrombolysis is good if given in time equals myocardium, all that stuff remains true to this day, despite the fact that today one would say, oh, well, that data definition was not of sufficient quality. It didn't match up to some standard. You don't have to be perfect to get reliable conclusions, but you do have to understand in what way are you imperfect, and to what extent is that going to matter to the type of conclusion you're trying to draw. So it's the avoidance of errors that matter to the decision making, not the avoidance of all errors.
To that. So one of the things that I think has been very valuable to the drug community with this collaboration is that we've accessed, as you saw, a large number of individual patient data, and it provides us an opportunity to actually find out what the quality is and how similar or different it is. And I guess a couple of things. One is that we go back and help the KOLs who need help with cleaning. We can help find systematic issues and help them solve them, give them tools so that it's kind of...
So you're offering janitorial services.
Absolutely, I like it. But I think the other thing that's really important to understand is, at least from our perspective, is that I'm not so sure it's always the case that aggregation is the best thing to do. I mean, sometimes it for sure is. But until you really understand how people run the assays. You know, a four-stair climb. Do you count the seconds? Do you count the capability? Do you allow them to the kids to hang on to the bars or not? All these things are not obvious when you're just looking at a database. So we've taken the principle of if there's an approach that's going to be useful to drug developers, it should be generalizable. So we discover in one and validate in a second. And maybe we'll learn that you can aggregate, but it's right now it's learning.
I like your answer, and you preempted a question I had for you based on the data you showed, which, you know, I've lived in a world of people who can find patterns in anything, and then you try to repeat it, and it's a different set of patterns. But you've answered the question, which is validate, and absolutely critical. Perry, I wonder if I can pick on you a little bit about, you know, one of the things that surprised me when I first started with big medical products companies was I assumed that the trials that they actually paid for themselves would have interchangeable data, but then I learned that almost no one does. What did you discover at GSK about the quality of the data when you actually tried to put it together?
The short answer, you know, it's a rhetorical question, very messy. But in defense of what GSK did, they undertook an exercise that goes back now probably 7 or so years to harmonize data, to have consistent standards within, to be able to cross-interrogate information, particularly from a safety point of view, which was a massive investment of everything from IT to informatics to analysis, which did make life easier. And much of the data from clinical trials available on the site immediately are in that format. Proximal to then, it was really, really challenging. There were paper records, even having just common standards and range of normal across the globe was incredibly problematic.
And our experience on polycystic kidney disease was that it was a lot of work because the data was not in very good shape, and it was only by being able to have big enough sample that we could get a subset that was useful for the qualification submission. But in other projects where we have data coming in predominantly from pharmaceutical companies, they may have different systems that they use within each company for structuring data, but there's good discipline and process for the running of those trials, so that has made that a little easier. But today, it is still too much work. It's very difficult to work through and do it correctly so that you're able to have some effective product at the end. And that's where going forward, we need to work on how do we make that whole process more efficient so that we don't have to deal with this. At the end of the day, you're still subject to how good are the data to begin with, no matter how well the trial may have been run. One of the approaches we've followed is to, at the beginning of every project, try to prioritize what are the data that would be of most value for a specific project and focus on those. And that has worked okay with some setbacks from time to time.
I appreciate those answers, and just a quick comment on this. A number of us who are working on the Precision Medicine Initiative have sort of come to the conclusion that we really need to objectify the discipline of curating data, because it's not going to go away in the short term. I think gradually we'll iterate towards better standards and terminology, but in the short term, I think if it's objectified and the discipline is better understood, people can plan for it and deal with it. Before we go to the mics, I want to do two things. Chuck, I want to ask you a question. Feel free to make a comment too, which I think you have, but now that you reacquainted me with fire instead of fur, I appreciate that. One of the things that's bothered me a lot, as some people know, is, but you implied that it's getting easy to do it. I found that health systems, and someone's already mentioned it here, are the least willing to share their information now compared to, let's say, companies. Is there some magic you've unlocked with FHIR that makes CEOs willing to actually share information? They seem to be hiding behind all the same things we used to hear from pharma about, their quality might be uncovered and people will misinterpret it and all this sort of thing.
Let me just comment very quickly on something that other panelists mentioned, which is the quality of data and how you collect it. I think the inherent issue, certainly when I was at AstraZeneca, was about accuracy of data collection. And so we'd collect data on patients at Duke, and they'd say, I've got the gripes. So we took it out of the hands of the principal investigator, and all the data on adverse events was coded in Wilmington, Delaware. They said, oh, we're getting more accurate data. I said, well, I think you're getting more consistent, less accurate data. So in all their wisdom, they took it out of the hands of Wilmington and put it in Alderley Park outside of Manchester, where I assure you no one knew what gripes was. Okay, having said that, in fact, I spoke at a meeting last week, and the former CEO at Sharp Health System, one of the 2 large health systems in Southern California, commented to me, because he's now retired, about their unwillingness to share data with Scripps Health. A lot of congressmen have this notion of information blocking, and it's the big EHR vendors who are doing it. No, it's the people who own the data who are doing it. Neither FHIR nor any policy will do it unless we create an environment in which there's some business case or incentive to do it. Otherwise, they'll continue to do that, which is, I've been competing with this entity for 20 years. Why should I give them any of my data now? So I think we, like Winnie the Pooh, we've found the enemy and he is us.
One quick point on that. I think the public largely brought about the opening up of pharma data, and I think we have a similar thing going on. The public needs to be more aware of the impact this is having. I know we have questions at both mics. I did want to give Kyle just a moment if you have anything you want to comment on or reflect on here.
Thank you. I did want to say one thing about how we're talking about combining data with this idea that we can put it together to essentially formulate an experiment we didn't do, a bigger trial, or test a hypothesis that we didn't exactly utilize any of the individual trials to elucidate. Oh, another thing that we in CDRH are envisioning the use of data from multiple sources is model development, and we're getting to the place now through the Medical Device Innovation Consortium where companies are willing to share data that they each have, both from model development and model validation, so that it's a different way of doing an experiment that you didn't actually do, which is to model it. And we've taken this so far that we were talking about Bayesian trials this morning. We're envisioning a world in which you can do a Bayesian trial where the prior data isn't real data. It's virtual data from a virtual clinical trial. And there are, very similar constructs to what you're used to thinking about when you do Bayesian trials, in terms of if the data that you actually collect is in line with your prior, then great, then things line up. And if they're mismatched, then you account for that accordingly. And we have a paper that we just submitted to the journal Clinical Trials on that. So there's just an additional richness to this topic that I just wanted to share with you that CDRH is partnering with industry around.
Fantastic. It's been fun to see all this work going on. We have let's go left mic and then right mic. Introduce yourself and fire away.
Hi, my name is Brian Corrigan from Pfizer. As an audience member today, the first talk and the last talk in this session seemed to give me sort of 2 different opinions of aggregating data and some of the dangers. And so in the first talk, we had 2 wonderful examples of where when the data was aggregated, the message was more or less the same, if I understood correctly. And then in the last talk, we saw an example of how important it was to sort of make sure you have a standard for the data in place a priori and collect the data carefully so you can use it again and again. And so the 2 didn't seem to quite jive. And in the panel discussion afterwards, we heard about in the big data case, if you understand what potential errors could occur and you can live with them, then that may be okay going into it. I guess my question is to the panel and to anyone in general. I'm thinking of a third example where, for example, in the US for climate change, for example, highly politicized topic. They changed, for example, in the collection of data, they changed from ship-based temperature measurements to buoy-based temperature measurements, and now to satellite-based temperature measurements. And as a result of that small change, they saw that the global temperature sort of flattened out. The climate change skeptics, even though the change was very small, glommed onto that, and it's become a highly politicized issue, sort of saying that, there is no climate change. And so that was not something, that error was not something that probably the NOAA scientists could sort of predict a priori that someone might use in that manner. And so I'm just sort of curious, from this whole session, is the overall message that it's okay to live with noisy data for big data, but we have to, in the cases of medium and small data, be more careful? I'm just want to make sure that we have the message from this particular session clear before we move on. Thanks.
So if I can comment a little bit. So from my perspective, I think you always want to have the best possible data, no matter whether you're dealing with small data sets or large data sets. Things go better. I focused in my talk more on the process of combining data sets, but as will be discussed later this afternoon, then you start this whole new phase of analyzing those data to understand what meaning may be present and what conclusions can be drawn. The approach we followed is tried to emphasize the ability to be able to continue to grow a resource and repurpose it as needed, because you may go down one particular path, find that there's some unsurmountable problem with the method that you were trying to have a scientific justification for. So you have to then back up, see if there's additional data that maybe help that effort, and then try a different analysis approach. But do it in a way where you can reuse some of that prior work instead of having to go create a whole new data project. Hopefully that answers some of your questions.
Yeah, so I mean, I think, first of all, to point out that what I was talking about was you're doing an experiment and what errors might occur in the experiment and what data might one use for that experiment. What some of my colleagues, particularly further down to my left, were talking about was you have done a bunch of experiments and now you're trying to bring them all together and analyze across them. And I want to just deal with one point, which is around standards and harmonization. At some point at which you've got the data coming up in front of someone who's going to analyze it, then having some form of standardization is extraordinarily helpful. That's why CDISC is there, is because it makes the FDA reviewer's life a hell of a lot easier if they are looking at the data in the same format every time they look at a trial data set. And that's just fine, and I have no problem with that. I guess what my comment on standardization was really, actually, if you go to the real world, there are different data standards. All the data in the real world could be standardized, but they'll be standardized in ways that you don't necessarily appreciate, approve of, haven't come across before, are perhaps legacy, are perhaps innovative and new, the same as you've got the same problems with both. So I think one has to cope with that variety of the forms of data that one comes in. The second point is about adjudication. And I guess my point there really is that one needs to consider what the impact of adjudication might be. And I actually think what adjudication means, it's slang that's commonly used by many of us in this room, regulators, pharma, academics, and so on. And what does it actually mean? Well, sometimes it means you're trying to distinguish an incident event versus a prevalent event. For example, is this a new cancer? Is this a recurrent cancer? Is, in the case of acute coronary syndrome, is this a new event or is it a continuation of the same event? As one of my colleagues once put it, is it raining again or is it still raining? That's adjudication. You're not changing the diagnostic code, but that's adjudication. The second...
Can I suggest it rains in England?
Yeah. The second is that you might be doing adjudication to simply try and identify more events that you might be missing. So you're trying to actually effectively reduce the number of false negatives, increase your power. And the third element is you might actually be saying about the false positives and trying to get rid of noise that way. And I suppose the fourth element is there are times when distinguishing between one type of event and another type of event that at one level look very similar, think stroke, actually is important, or death, is actually very important. And so if you're doing a study of warfarin, distinguishing hemorrhagic stroke from ischemic stroke, they're both stroke, is critical. If you're doing a study of antihypertensives, it probably doesn't matter much. People say, well, all-cause mortality is the ultimate endpoint. Well, yes, it's the last one that happens generally, but it actually has enormous problems because you distinguish, you mix up different events which might have different treatment effects and might appear at different proportions in different populations. So when you go from the old to the young, you may get very different distinctions. So those sorts of adjudication, that fine-grained classification characterization might actually be important. You've got to think what you're trying to do with the data in order to then interpret the result.
Isn't the main lesson here, there's no substitute for experienced, highly qualified people who look at a problem and design the analysis to fit the quality of the data and the question. But Perry?
I agree. I was just going to add a little bit of a flavor to that. I think it depends what's to be adjudicated, and to the extent to which it's adjudication of that which is interpreted by, say, pathologists or radiologists, to pick on two. I think that's one kind of adjudication. Whether adjudicating a lab value inside or outside what's normal, notwithstanding the harmonization, I think is another.
Rob, if I may, just very finally on this, I think there's a distinction between the traditional, what I do on a Wednesday morning when I see one patient, where diagnostic accuracy is what it's all about. I need to be precise in this one person. And when I do clinical trials, which is I want a precise estimate of the treatment effect. So sort of diagnostic accuracy studies are not what I'm interested in when I'm looking at the trial. I'm interested in the effect and the impact on the effect of my assessment of the effect of the treatment.
Always find when I'm around Oxford people, I'd like to have another 6 hours to argue more, but we probably need to get Alistair.
Yeah, I'd like to challenge something that we've sort of seemed to have accepted without discussion, and that's who owns the data. The assumption seems to have become pervasive that the data is owned by the hospital or the data is owned by the drug company. I would suggest to you the data is owned by the patient, and it's kind of like my bank. When I put money in the bank, as a Scotsman, I'm pretty sure the bank doesn't own my money. And, I certainly hope not. And so this attitude that we've developed that somehow because you go into a hospital and have tests done that I pay for, by the way, and that are put in their record, that they're somehow the owner of that, is a big problem. Now, this is not just a semantic issue. It seems to me that if we move towards the issue where we recognize the data are actually owned by the patient, then the person who we should be seeking permission from to share data is the patient. And perhaps more importantly, the person who should be assuming that data are shared, particularly amongst hospitals, for example, is the patient. I mean, would you put your money in a bank where you didn't know whether the bank was secure? Probably not. Would you go into a hospital that refused to share its outcome data with other places if that was public? Probably not. So I think there's an issue here that goes beyond semantics that would make a fundamental difference to this discussion if we recalibrated it.
Susan?
I couldn't agree with you more. And you see it in spades working in a rare disease. It's something that I think the academics are actually very sensitive to. One of the reasons that it's been very difficult to persuade the academicians, the clinical KOLs, to share data, some of whom have never shared data before, is that they feel a responsibility to the patients to not use the information for one drug company's benefit over another. So they need a forum in which they can share, make sure that there's a third-party adjudication that it's okay. So the consent, even with data that we have, if we want to do something that is not quite exactly inside of the consent, we go back, or they go back to reconsent. There is also an example that I have heard of. Well, yeah, I understand. So there's research and then there's on the borders of research. And usually they're consented for research purposes. And sometimes it might be answering regulators. Is that a research purpose?
I mean, my comment here is watch the space closely right now, because if you look at the proposed common rule that's now on the street, I wouldn't urge that all of you read all 595 pages, but there's a lot in there in the precision medicine initiative that Republicans and Democrats and President alike are all in favor of, remarkably enough. It's going to very much push the idea of broad consent and volunteering data and notifying the public that your information actually does belong to you. It's going to be a very interesting time. Alistair, do you have another follow-up comment before we?
That this is a somewhat generational issue. You know, there are those of us who grew up in the HIV era in which patient confidentiality became critical. Then there's our children who share information multiple times a day that we would never even have considered telling people 20 years ago or whatever. And then there's when you actually go out and poll people and ask them how, if they're willing to have their DNA saved for genetic analysis later, the vast majority in the real world, in the Vanderbilt example, have said yes. And so I think, you know, those of us sitting around here who tend to be of one generation see this from a different perspective from our children, and we need to adapt to that.
Urge that everybody go through a Senate confirmation proceeding, and then you'll know what data sharing is really all about. Steve has reminded me that we're running out of time, so if we could have sort of rapid fire. We have 2 questions. If you could make them quick, and the leading panelist, whoever wants to be, could comment.
Okay, sure. Once again, my name is Mark Gorman, and I'm a cancer survivor and patient advocate. So I actually had 2, but you got to my second one just now. I will say, in the circles that I travel in, you know, patients are all over this idea of we own the data and we should actually control its portability. Now, you know, I'm not totally convinced that totally open is okay, but you know, the larger context I want to reintroduce here, because it has come up over and over again over the years when I've been in sessions like this, is that a big part of the problem, and this isn't just the FDA as a regulator, but payers and clinicians, you know, we don't know enough about what's called the natural history of disease or the natural history of being well. And if that were understood better, many of the kinds of questions that we've been talking about today wouldn't necessarily be so vexing. But having access to data to be able to do that would be helpful. And the Duchenne's case reminded me of that. And I know that's the argument behind the Alzheimer's sharing example. So I just want to get that idea back. We were encouraged this morning to put things into the record. And I think that's a really, really important concept that we shouldn't lose sight of.
The last
I'll just do very quickly. We talked a lot about the big data in the context of infrastructure and large data sets, but I think we didn't touch upon, and maybe this would be a topic for another workshop, is really of the need to look at the methodologies and analytics behind these large data sets. It's not only about large data, but it's about the fact that you can't just apply the traditional methodological approaches. There is that need of simultaneous kind of algorithmic and inferential approaches and machine learning and all of these other aspects that make big data so appealing. So it's really not only about having huge data sets. That's all.
Thanks for that, and I can assure you the FDA is very concerned about that, so much more to come.